started. Debi, with your background in business, and Tarina as a bench scientist, how did you both end up at the Michael J. Fox Foundation?
I did not come to Parkinson's with any personal connection. I didn't know anybody with Parkinson's until I met Michael, and I was a little distracted by the fact it was Michael as opposed to the fact that he had Parkinson's. And I came from a background, and my first career was in business. I worked on Wall Street. And I had been at a great firm and great colleagues and great clients and customers, 9 years at Goldman Sachs. But I had been in this time where I loved it, but I didn't love it. And it's hard work, and I wasn't afraid of hard work, but I really only wanted to do such hard work if I felt like there was just a little bit more that was going to be good for the world out of my efforts. And I found my way, I found this tug that I really wanted to find my way to the nonprofit sector. And I bet that I felt the way that many of you do, which is there is no end to the number of noble causes. So I didn't really find myself thinking, oh, I must go to this area or that area. What I started and what I really brought to my kind of transition was I had a real passion for how philanthropy capital should be used. So I sound like a business person when I start to talk about philanthropy, but I really felt like, wow, all these noble missions, but it seems like very few people move the dial. And so I was really trying to figure out how could I go into the nonprofit sector and really help shift a gear so that an organization could be particularly impactful towards their mission. I didn't pick anything easy. Parkinson's is not simple, but I really did come into this idea that if I could go and start, put my energies to work and think about how do you introduce change, how do you think about impact in the nonprofit sector, I'd be very satisfied. And so I got connected to Michael through 10 degrees of separation, and it was kismet. So I'll tell you a quick story. We met on Thursday and in this interview, and I was all nervous. And I thought, wow, I had no idea how much I wanted to take a shot at something like building something from scratch. And the interview's over, and I walk out, and even at Goldman, I'd done a lot of interviewing and stuff, and so I kind of knew what I needed to leave on the table. I walk out, and I'm thinking, I did pretty well, but I'm probably not at all what they're looking for. I mean, I had no medicine background, no nonprofit experience. I had no, except for as a volunteer, and I had no scientific background. But I thought, I had a little swagger, and I go down to the elevator to go downstairs, and he walks up and stands next to me, and he keeps talking. I'm like, shoot, this is not over. So we go down in the elevator together. We walk outside on Park Avenue. We stood out there for another 45 minutes talking. And I thought, he is a prince. He's just what you think he's going to be. And I was so motivated. And he says, I'm calling you later. I want you to do this. And I said, if you call me later, I'm totally doing this. And he says, I feel really bad, but I have to go upstairs. I'm supposed to interview more people. I said, you go. And so he called me that night, and he offered me the job. That was Thursday. I started on Monday. It was just like that. And I had only heard about the job the prior Sunday, so almost just about a week. But that was very transformative for me. And I think for him, we had a kismet about what we wanted to accomplish. And even though we didn't know much, we knew what we didn't want to be, and we knew that it would take hard work to do what we want to do. So I was really drawn into the sector by the opportunity to build something from scratch with this idea of impact in mind. And I was no idiot. If you're going to start from scratch, you're much better off starting with someone like Michael. Because one of the things I also saw with most nonprofits is even if you have a very powerful idea, it's usually an organization with fewer than 7 employees and a budget less than $1 million. Scaling good ideas is hard. And I knew right away, Michael Fox, really good chance of scaling. So that was really my gut. That's where I was going from. And when I look at what we are today, it was unimaginable, I have to say. But I couldn't be prouder, and I know he feels the same way. We didn't know where we're headed and what was going to be. We just knew we were going to put our heads down, work hard, and put our arms around anybody who wanted to walk with us. And it's been an extraordinary journey so far.
So I ended up at the Michael J. Fox Foundation, which was my dream job when I first started studying Parkinson's disease. I was actually in college. I was pre-med in college and thought I wanted to go the regular doctor route. But I did some research on the side, some basic science research, and really, really liked it. And then I, like all type A overachieving people, I overanalyzed that and thought, I'm not sure. I wanted to go to med school my whole life. Let me really dig into this research. So I did a fellowship after I got out of college and happened into a Parkinson's lab, and just really was fascinated by the puzzle, the scientific puzzle that is Parkinson's disease, by the brilliant researchers who were working hard to try to move the science forward, and then the patients who I met along the way. I was fortunate enough to be at a graduate school that had a large movement disorder clinic. This was in Dallas. So all of that reinforced my choice. I went to the University of Pittsburgh Medical Center for a postdoc fellowship, and started thinking about what I wanted to do when I grew up as a scientist. And the opportunity at the Michael J. Fox Foundation came across my network, and I thought it was just really nice synergy. My graduate studies were funded by a grant from the Michael J. Fox Foundation, so my training was actually part of that whole process. And when I went up to the offices and met everybody, it was just a wonderful fit, and it's just great to be part of just a collection of brilliant people from all sides. I mean, everybody up there is just passionate, and it's just wonderful to be part of that.
So it turns out most disease charities actually aren't focused on research. I mean, some a little bit, but when you look at the programmatic efforts of most disease charities, they kind of grew up a little differently than we do. They start in communities, grassroots efforts, and most of the effort is focused on providing services in the community. And so it's really a little bit more towards care and coping and living with disease challenges and maybe advocacy. Those are common features of disease charities. And Michael, when he was getting his start, one of the first things he was asked to do, given that he was Michael, was to go testify before Congress about the doubling of the NIH budget. So his immediate immersion was around science. And to him, it's not that he didn't think that services were important, but I think he felt he could really galvanize significant funds. His hope was that he could do that to really speed research, because he was hearing a clear message from the scientific community. The science is ahead of the money. Takeaways. Science is ahead of the money. That's where I'm going to focus. And there were already a couple of existing Parkinson's groups who were doing the other thing. They had chapters and grassroots efforts, and he thought he could, and he actually went around and talked to all those groups before he contemplated starting his own, and he realized nobody's really focused on research. And by the way, it's not just in the Parkinson's field, that's common across all disease charities. And so that was his focus, and my belief is, you tell me what your mission is, and that's what we're going to be focused on, because I was really thinking, how do we move the dial given that mission? And so research it was. And now we had to start from somewhere. And I think for me, one of the biggest shifts was on Thursday when I said yes to that job, I thought we're going to be funding research. That's what my takeaway. And by the end of the first week, I started to realize, literally, by the way, this predates Google. Like, you couldn't just go online and say, how much research is funded in Parkinson's disease? Like, and you really couldn't figure that much out very quickly, but I started to ask questions, and I realized there's actually a lot of research funded. I mean, there can always be more. There's no question. You could always have more money for research. What I started to realize is that given how much money is used, why aren't we making more progress? And that zeroed me in on this idea that we're not just here to fund research for research's sake. We have a specific goal. We're trying to drive treatments to the patient community. And that really was an immediate and quick shift for me. We're not funding research, well, yes, we are, but we are going to fund research and direct our investments in research in a way that will speed drug development. In Parkinson's disease, well, in any kind of central nervous system disorder, which is where Parkinson's lives in the drug development world, it usually takes, sadly, at least 30 years to go from aha moment to drugstore shelf. And it can cost probably $2 billion in today's money. And so, yes, a lot of money is needed, but I very quickly, and the early board members, we all really zeroed in on this idea that it's not how much money you spend, but how you spend it and what you focus it on. And for us, the early decisions were around the art, which I think it is an art. What's the difference between interesting scientific questions? There's no end to the number of interesting questions. And the questions that are relevant for drug development, much smaller target. And so we started to prioritize that. And then I realized very quickly that we would need, not me, a business person trying to decide which science was best, and not an external group of people that would come in once every 6 months and we'd say how much money we have and then they'd hand us a pile of checks to sign. I really wanted to make sure, because I think the traditional group of scientists would fund all the interesting science. It's not common to really target this subset of science that's focused on drug development. But if you have the patient in mind, you want to really target that subset. And the NIH, for instance, which is the most amazing funding apparatus for science in the world, they aren't focused on drug development. They're focused on science for science's sake, which is an underpinning of discovery that is undeniable. But on top of that, what you want to do is figure out, now what? Given these fundamental findings, given this new knowledge, given this incremental bit and piece, what are we going to do to now race that thing through the gauntlet and to the patient community? And so this was kind of how we were crystallizing where we are going to go. And it focused on things like, what kind of risk are we going to take with our capital? How are we going to make decisions? Right out of the gates, started to think about hiring our own PhDs as opposed to outsourcing that to, I mean, we connect with other PhDs all year long, hundreds and hundreds, from academia, biotech, and pharma. But we still integrate that. Trina and her team, our collective group of scientists are really running a portfolio of the kind of scientific investments you need to make to get things over the goal line. So I think I've spoken too long, but I think that's, those were some of our early, how we were shaping what we were going to do. And I think keeping those patient values in mind, which is what's relevant for patients. It's new and better treatments. What matters a lot to patients? Urgency. Like 30 years, that's unacceptable. Now, biology's hard, you can only do so much. But I believe from day one, and I see it today, there are so many things that you can do faster and smarter because nobody else was doing them. It's not that we're doing them better, nobody was doing them. And so we've been able to take kind of haphazard pattern of science and progress in a specific area like Parkinson's and make it far more strategic and impactful and milestone driven and kind of a portfolio of things. We don't bet on one horse. We're happy to place many bets because most things are going to fail. And so that approach was really at the start, and it has grown into today really probably the single most knowledgeable group of people in the world about Parkinson's disease, science, and drug development that knowledge resides at the Fox Foundation, which is something we're incredibly proud of, but it also means it's changing how things are moving in terms of getting treatments to patients.
Thank you. And from the outset, you and Michael really, again, decided to say, let's take a unique approach to this, let's tackle the problem in a way that nobody else has. Can you tell me a little bit about why you decided not to have an endowment? That's another unique part of the foundation.
Yeah, so a lot of service-based organizations, some have endowments, some don't. Big funders of science often have big endowments. And we just kind of felt like, I came from a place where use of capital was kind of my rubric. And we just felt like, why would we raise a lot of money to put it in the bank if there is smart science in front of us to do? And given the cost of science and the high failure rates, you really are not going to run out of ideas, even if they're the pinpoint of the stuff that's going to drive drug development. And so that is an uncommon approach for a group of our size and our nature to big science funders, but we do not have an endowment, and I'd say I guess it's a little bit different. I'd say we value capital in a very specific way. So it's about the money coming in and the money going out. So in the course, the half-life of a dollar at the Fox Foundation is probably negative. We've probably committed it before we've raised it. And even though, the timing can be a little off, we will raise in a given year. This year, this is a crazy amount of money when I say it. We're trying to raise at least $100 million. We've already we haven't raised anywhere near $100 million yet, and we got 3 months to go. But we've already committed almost $70 million to new grants. And so we will, as we get towards the end of the year, we're going to get close to that number. We won't spend the millions of dollars that come in the last 3 weeks of the year. But those will be spent in January. So if I look back at the last 12 months, we've spent $120 million in commitments on new science. So that rolling distribution of funds is about urgency. And then I guess another thing is, maybe it's because my background and a lot of the board members come from the finance world, we are very interested in financial accountability, and 89 cents on the dollar at the Fox Foundation goes to the program. And it helps that the denominator's big because science is expensive, lots of PhDs, they do thinking, it's not their own labs. But this is, we're proud of making sure that when we use a dollar, we use it smartly. And so those are some of the other things that we were just committed to on day one. And I think that it helps people understand that we're on the same journey that they are. I mean, if this is just, it's our passion and it's our purpose, and this is other people's money.
Exactly. Thank you so much. And I know one of the things that we are so proud of and is important to us is measuring progress. And in your time with the foundation, can you talk a little bit about how maybe the spectrum has changed in terms of our engagement in the scientific community across academics, biotechs, pharma?
Yeah, I think, there's some big things that have changed that are kind of about medicine and science, specifically in Parkinson's disease. I'd say one of the biggest things that's different, when I started my job, probably the first question I asked is, how does someone get Parkinson's disease? And this is only 15, 16 years ago. The answer was, it's environmental. And in fact, I said, well, I've met some families where more than one person has Parkinson's. Yeah, well, that can still be environmental. People share, families share occupations, they share environmental exposures like well water and working on farms. And we can't explain very much about what we think causes Parkinson's, but all of the signs point to environment. That is a big change because now I think we've implicated over 20 genes that are either causing or high risk factors for Parkinson's disease. That is one thing that has really transformed, probably two things. It's transformed potential targets for drug development, and it's also helped connect the biology under something else that I think is a big shift in Parkinson's, which is the idea that it was only a dopamine problem. All the medications at the time we started were really focused on replacing dopamine. Something's gone wrong with the cells in the brain that produce dopamine. Let's just put that dopamine back and we're going to be fine. So going beyond dopamine and starting to appreciate, no, this is more than just some of these motor features. There are a lot of things going on in Parkinson's that patients could always describe, but doctors and scientists kind of disregarded because they couldn't connect the dots. And now, between a better understanding of what Parkinson's encompasses, all the motor features and the non-motor features, and then when you put that side by side with what we've learned from these new potential causes of Parkinson's, you can start to connect the dots also on some of the biology, and it starts to make more sense. Our picture of Parkinson's disease has changed pretty dramatically. And that tees us up very well for faster progress. I'd say the second thing that's remarkably different, and even in speaking with some of you last night, the folks mentioned this, the awareness about what Parkinson's is a whole new world. And I think, I personally can appreciate the role Michael Fox has played in that, but I think it's everywhere. And Parkinson's is a disease that many people kind of internalize, and they kind of turn in. And people, Michael Fox kept his diagnosis secret for 7 years. A lot of people still keep their diagnosis secret, but it's rare now that they don't kind of find some support and some comfort in sharing earlier. And a lot more people understand when they say this is what they have. Oh, I know what that is. I know what Parkinson's is. And I think that has been good for patients, but it also has helped speed progress, because as patients are more engaged, they're doing things like coming out together and getting better educated. They are funding research, and they're participating in research. And these things all add up to making a big difference. So the role that the patient is playing has gone from hiding and quiet and passive to engaged and more active. There's a lot of work to do there. We still do not know most Parkinson's patients. They're hiding. And they're quiet. But a lot more are present, and that can make a big difference as well in our progress.
Well, and I think just to build on that, Debbie was saying how transformative and just a decade ago. We didn't have the idea about the connection of genetics to Parkinson's disease. But from the science perspective, I wasn't at the foundation when it started, but that was when I was beginning, I was in college and beginning my scientific career. And I look at where we were then and where we are now, and the capacity to scale and actually move things faster, in addition to the work that the foundation has done, science has just started to move that direction. There's more collaboration between small labs, biotech companies, which are medium-sized, large pharma groups. Everyone's kind of, getting with the program that it doesn't make sense for everyone to work in parallel, in competition on the same tough problems. That's a redundant approach, and most scientists really just want to, move the discovery forward and get it into the public domain, but that's easier to do when you're talking to each other. I mean, there were times when I was in grad school that the best conversations and collaborations happened around the coffee pot, not because we were in meeting rooms and brainstorming. So I think that scientists have gotten more savvy about talking to each other, and outside groups and the brokering and bridge building that the foundation does has helped with that as well.
Great, thank you. And now let's switch gears to the state of research today. Our greatest unmet need in Parkinson's is, of course, a cure, which I think that we, again, with Parkinson's can define as the stopping or slowing of the progression of the disease. And one strategy that we're very focused is around a protein called alpha-synuclein. Serena, I'm going to ask you a few questions around alpha-synuclein, and hopefully we can explain what alpha-synuclein is and why we're so excited about it. So first and foremost, what do we know about alpha-synuclein and why are researchers so interested in it?
Right. So alpha-synuclein was one of those first genes that was linked to familial or inherited Parkinson's disease. We learned very important things, firstly, about alpha-synuclein that told us more about this, unlocking some of these secrets of Parkinson's. So alpha-synuclein is both, it has the name for the DNA that turns into the protein, and we learned that some of the mutations caused that protein to clump up and misbehave. And what's interesting is sometimes that mutation could change the protein, but other times the mutation was just too much of the normal protein. And then scientists, as they started investigating, once they knew how to see alpha-synuclein in a cell and in the brain, they started looking at patients who didn't have mutations in alpha-synuclein. They had environmental or other causes of Parkinson's disease, and they started seeing that alpha-synuclein was clumping up and misbehaving in those cases that were not linked to alpha-synuclein mutations. So that was a big light bulb moment for the field.
So suddenly alpha-synuclein is the first gene that's discovered, and you can link it to cause. And still no other culprit has been characterized quite this way. Trina's explaining how this misfolded alpha-synuclein, though, is in the brain of every Parkinson's patient, whether they have the gene mutation or not. And those are things that we started to understand through the study of pathology in Parkinson's. And many people will have known of dopamine, and they'll have heard of Lewy bodies. Folks have heard of Lewy bodies, which are the clumps that are in those dying dopamine cells. Once the protein alpha-synuclein had been discovered, it's resolved a mystery. What is this Lewy body made of? And ding, 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 it was misfolded alpha-synuclein. So now there's something that we can link to cause and to pathology. And those two links immediately catapulted alpha-synuclein to the front of the list in terms of a target that clearly is playing a role in pretty much every Parkinson's patient. Independent of whether they had a mutation, they had this misfolded protein in their brain.
Yeah, exactly. And that's what made it just like Debbie said, it took it to the front of the list. And so then the scientists had to look at what is it that causes it to start misfolding? Can we stop that process? Once it's misfolded, can we clean it up? Just turn on this. Your cells and your neurons have wonderful capacities to deal with stresses every day. But why can't they deal with that extra stress, that extra burden of the misfolded alpha-synuclein? So now it's an active area of research across the globe to try to find ways to lower alpha-synuclein levels outright so that you can bring that threshold down so it doesn't start misfolding. Once it is misfolded, can you kind of coach it back to folding properly? Can you clean up those accumulations of it that are inside the Lewy bodies? So all of those require a very pointed approach, and each one is different. You're not going to find one drug it's unlikely to find one drug that can do all of those different steps. And that's what Debbie was saying. This is why we have multiple shots on goal. Alpha-synuclein is the prize, but we have funded and science is researching many, many, many different ways of targeting alpha-synuclein. And we'll talk later about how some of the drug trials are targeting it as well.
Well, great. So on that topic, you're describing a few of the different ways. There's many approaches that the scientific field is looking at to attack alpha-synuclein, and there are 4 projects in human testing today. So where are we in the field with alpha-synuclein testing?
Right. So right now, the areas that are most advanced for targeting alpha-synuclein use kind of a vaccine approach. So similar to the way, we're all coming up on flu season, coming up now. The way the flu vaccine works is by giving your body a small little bit of, there's different ways that vaccine approach can be manipulated, and those are 2 of the most advanced approaches for alpha-synuclein right now. And then one of the things that we're doing at the foundation that I think is very important when it comes to alpha-synuclein is looking at finding an imaging tracer. So many of you are familiar with the DAT scan that uses a specific way of looking at the dopamine transporter. We're trying to find an imaging marker for alpha-synuclein so that you can actually see whether or not these vaccine approaches are changing alpha-synuclein levels in the brain. You can use that for earlier diagnosis. You could use it in clinical trials. It would be absolutely game-changing to get an imaging tracer for alpha-synuclein, and that's an active area of research that the foundation is pursuing.
So just to put this in context, for instance, in a given year, well, probably for the alpha-synuclein was identified probably in 1998. Does that sound right? But it didn't really get into the scientific mainstream and in conversations probably until the early 2000s. By the way, that period alone now happens much faster. People jump on new concepts much more quickly. But I'd say since early 2000s, in the last 10 years, we've spent probably, on average, $12 million to $15 million a year just going after alpha-synuclein. We bring together consortiums to tackle the imaging issue. We have some groups where we're coming in and going after clearance, one of those strategies to how do you get rid of it. And we're working with academics, with small biotechs, and even big pharma partners when they get involved. And we just bring people together and say, okay, given what we know today, what's the next smartest thing to do? And I'd say one of the things that has shifted, and particularly as our organization has scaled, and to me it's one of the biggest impacts, as we've scaled, we don't have to do one thing at a time. And in the early days, we could only do one thing at a time. And now to be able to put to work a significant amount of resources year after year on alpha-synuclein, you can see how things are moving. And these, I think there are now 5 maybe alpha-synuclein programs that are in human testing. And we've in 4. Yeah, 5 maybe someone's starting.
Right.
Yes.
They're enrolling.
They're enrolling, yeah. So, but we've really been involved in all of them in some form or fashion. And often we're a funder in the early stages before whoever's taking it into the clinic today, before they bought it. So we might have worked with the lab in the very early stages, and we put our capital to work in a way that where our goal is to de-risk the idea enough so that a big pharmaceutical company will come and buy that and get it into the final phases of testing. And I think this is kind of important. It's a little counterintuitive. I mentioned before how much taking one idea, the distance costs. It can be close to $2 billion. We actually don't know how much it costs to get a disease-modifying drug in Parkinson's because we don't have one yet. So we don't really know how much that costs. But the phase 3 trial for disease-modifying therapy in Parkinson's, it's pretty clear it's going to cost $500 million. We have to have a lot of kicking PD parties to raise $500 million, and we don't want to just, you can do it. So we are not under a false pretense that the Fox Foundation is going to take anything over the goal line. And I actually think this is important. We're very sober about the role we play. Because if we're too Pollyanna-esque about this, we won't be smart about how we use our money. So our goal is not to think, oh, we're going to fund that phase 3. By the way, we're going to do everything we can to help a phase 3 get funded, but our money's never going to get that done. So we use our money in the earlier stages. By the way, drug development is a little bit like a big game of Texas Hold'em. The early stages don't cost that much, and you get a little bit of information, and you keep making your bets. But by the time you're into phase 1, that's a $60 million trial. Phase 2 is a $200 to $300 million trial often. And so what we want to do is the preclinical and the early clinical stages. Preclinical, we might be 100% of the funder. And we're given the couple $100,000 and then good data, we'll put down a couple of million, and this stuff keeps going. By the time something's in phase 1, we might put several million into it, but they need other investors. And so we're trying to put the data around the idea in those early stages when we're the only one who will take that risk, because we're the only one that's putting that many bets in Parkinson's, so that the life science venture capitalists and the big pharma business development folks will say, this is one we need to keep our eye on. And in fact, what we know now is that companies who are trying to move some of these programs along in the really early stages, when they get funding from us, they know that that makes a big difference to those investors, because they know that we have really done the work to vet it. We have the context of all the things that are going on. We help shape we contribute to the thinking around the science so that the right questions are asked and answers to deliver the data package that the investors are going to look for. And so this idea of being able to do as many of these things at once, to us, is one of the main ways to keep progress moving. And the signs in alpha-synuclein are significant. Although it's not, I mean, it makes it sound like that's our only bet, and it really isn't. I'm curious how you would describe this. I'm going to say how I think it is. The scientists kind of are always looking at the state of science, and periodically, and it's at least once a year, they kind of sit back and it's like handicapping horses. I have a lot of sports analogies today, or betting analogies, I guess. And at any point in time, someone's pulling ahead. By the way, failure, kind of sideways outcomes, someone pulls back. And they're kind of looking at the state of the field, and then they're going to deploy their resources and kind of say, this one is going to get the $15 million investment. We have another top genetic target called LRRK2. It's not in human testing yet, but it's getting close. We've spent $12 million a year on LRRK2 for the last 8 years. Right, so LRRK2 is another one. And it's interesting, it sounded at first like LRRK2 might only be helpful for folks who have a LRRK2 mutation, which it is the most common mutation. About 1.5% of all Parkinson's patients carry a LRRK2 mutated gene. But in some subsets of Parkinson's patient community, for instance, Ashkenazi Jewish patients, it might be as high as 20 to 40%. Carry this LRRK2 gene. And early signs were we couldn't really connect the biology, so we weren't quite clear. Maybe just going to help LRRK2 patients, but it's very interesting. We're just getting new data that's saying, nope, we're seeing how it may find its way into that alpha-synuclein pathway. Looks like it's going to, and LRRK2 patients look like a standard classic Parkinson's patient. There's a lot of reason to believe that any LRRK2 drug is going to be very beneficial, could be beneficial to all Parkinson's patients. So, we're placing bets, and we have some emerging targets at any point in time, but the whole goal is to say, okay, now what? Where's everybody? What do we do next? And as we pace our fundraising, we kind of have shovel-ready science to keep things moving.
Yeah, I would say like when the scientists get together, I call it chasing rainbows. We just look at the scientific problem, as Debbie was saying, and there's sometimes that we can look at it and say, you know what, for example, a lot of people are in the alpha-synuclein space right now, and we've pushed really hard to get that to move forward. So where can we make the biggest impact? And so as for some of the new emerging targets that are coming out, LRRK2 is very exciting. So is the protein Parkin. It was one of the first, right after alpha-synuclein was discovered in 1997, mutations in Parkin were discovered. And what's interesting about Parkin is it's a very, very rare mutation. If you look at the genetics of it, it's interesting, but it doesn't look like it would be big impact. But now we're thinking that sometimes for things like alpha-synuclein, it looks like kind of shutting down alpha-synuclein is the way to go. And Parkin is the opposite. You want to turn it up. So that takes a whole different process. I mean, the way a scientist would approach the problem of shutting something down is completely unique to the way that we would approach turning a different target on. And so it's great, though, to be a scientist at the foundation, to have, and our desks are all close together. I mean, the nerdy conversations we have constantly, is just so stimulating and exciting. And then we also have connections to, the Parkinson's researchers in, our local community, across the country, across the globe. But we really do chase the rainbows. And then we have these brilliant business strategists who, rein us in and say, okay, if you can only do one of those right now, which one is it? But then I think that's also what Debbie was saying, when we kind of run a little bit behind, and we've caught up with the money to the science, but the science is still way ahead of the money. And we, on any given day, if I woke up tomorrow and had, $5 million added to my portfolio, I know exactly what I would do.
That's great. And if some of you may have been at last year's small roundtable that we did, and you may recall that I was highlighting, hey, this part's important, this is exciting. And so as I'm listening to you, again, the message is, this is exciting. In the area of disease-modifying approaches, we have so many novel targets, more than we've ever had before. There's a lot of work going on with alpha-synuclein, as we just heard. Again, more shots on goal, more horses in the race. Use any analogy you want, but it's good news. There's a lot of progress being made.
And some of these are already in human
Exactly.
testing, which I think is key.
And so there's a lot to be excited and hopeful about. So one of the most important things as we search for the cure, a disease-modifying therapy, is a biomarker, which I think would make things move more quickly. Tarina, do you mind briefly defining biomarker for us?
Sure. So
Thank you.
we are still trying to find the ideal biomarker for Parkinson's disease. So it's hard to really identify or define it within Parkinson's because we're still looking for it. So I'll go outside of Parkinson's disease to really help you grasp the significance of what this is to any disease. A really good example is high blood pressure. So high blood pressure is, something that many people don't even know they have it until they go to their doctor a few times and they test and they're like, your blood pressure's running high. That's a risk for a couple of different diseases. It's a risk for heart attack, it's a risk for stroke. So depending on how high it is, your doctor may say, or if you want to get really, really, you want to go back and work on your diet and work on your exercise, there's two choices. You could do diet and exercise, or you can take the medication. Say the doctor gives you a chance to go do your diet and exercise, you come back in 3 months, but your blood pressure's still high. Then they're going to put you on a blood pressure medicine. And then next time you come back, your blood pressure's back low. So in this analogy, high blood pressure is the biomarker. It is something that indicates it either indicates the disease is present or that the disease is progressing. And in this analogy, by reducing that biomarker, it also tells you that your therapy is working. So for Parkinson's disease, we're looking for the golden ticket biomarker. We're looking for something that may be going up in the disease before people start having symptoms, something that you could test in a imaging analogy or in a blood test or maybe a spinal tap. But we're looking for a biomarker that we could see the disease sooner. And if we have that biomarker, we can then also use that in clinical trials to tell us if these different approaches that we talked about, for LRRK2, alpha-synuclein, or Parkin, is that changing this biomarker? And I really think that would be one of the really important areas that would move Parkinson's therapies very quickly. So we have a lot of different biomarkers that are candidates. It could, like I mentioned, it could either be through brain imaging or it could be through blood screening. But we know that Parkinson's disease is a disease of the central nervous system, and that makes it very challenging to find a biomarker because we hope we can find something in the blood that tells us what's happening in the brain, but that's really risky. That's connecting a lot of disparate dots. It can happen, and we're trying, but it's asking a lot of a biomarker to just be something that you can test in. I actually don't even need it. It wants to stay there.
Yeah, that's fine.
That's good. Thank you. But that's where I think that efforts in finding a biomarker are very key, and there's a lot of active research. We have a very big biomarker study at the foundation. I can actually let Debbie explain how it was, because that was, talk about early vision. That was getting ahead of the curve.
Yeah, so I mentioned before, we kind of soberly realized we're never going to take anything over the goal line because we're never going to have the capital to put to work in a phase 3 trial. Another thing that we realized early is without these biomarkers, some kind of objective measure, you were really asking a lot of anybody who was going to put $500 million into a trial. Before you would measure cholesterol or blood pressure, the way we tested heart disease treatments was we gave them to 30,000 men and followed them for 10 years. Nobody can afford to do anything like that anymore, right? So now what you do is you give 2,000 people something for 6 months, and you read their blood pressure and their cholesterol levels, and you just say, yeah, we're onto something. So if we can't collapse the number of people and the time we need to follow them to understand whether drugs are working, chances are nobody's going to put the money on the table for a $500 million bet. There are a lot of people in this room who are in real estate. Can you imagine if you bet $500 million on something and your outcome was 0 return? Now, you could put a lot of money into a big building project, build new stadium, and it may not be the best stadium, but at least you have a stadium. Like in drug development, a lot of people put $500 million in Alzheimer's. Now the average trial, phase 2 trial, is $1 billion bet. They're syndicating this. Pharmaceutical companies are rich, and they're still syndicating these bets because it's a binary outcome. These are really hard things to get people to imagine. And I know, listen, I would love nothing more than to be arguing more with pharmaceutical companies about the cost of drugs. We're not there yet. But I can appreciate that we ask a lot of trying to figure out how to get to treatments and what it's going to cost to get there. And so these objective biomarkers can play such a role. So we realized, well, who has an incentive to develop the biomarkers? Because it's just like developing a therapy. It costs as much, it takes a long time, you have to test them in people and validate them. And this became such a clear clearance call that we needed biomarkers, and we brought a group together of pharmaceutical companies and academic labs, and we kind of said, what can we do to make this happen? And we launched this study now, we started planning probably 10 years ago for this study called the Parkinson's Progression Marker Initiative, PPMI, and it is now it's like a flagship effort in Parkinson's, actually across many diseases. There are only a few diseases that have anything like this, and we have led this charge in Parkinson's. And ironically, we spend just as money in biomarkers as we do in alpha-synuclein. But we're the only ones who really have an incentive. Because one thing about biomarker is, it's kind of like the tragedy of the commons. Everybody wants one, but no one wants to pay for it. And so I remember there's a neuroscience venture capitalist in Boston. His voice always rings in my ear, and he says, if you did nothing but find a biomarker for Parkinson's, for progression of Parkinson's disease, it's a home run. It transforms the entire investment appetite for Parkinson's disease. I said, well, we'll be on that. And we have. And so we still don't have the golden ticket, but now we can measure a lot more things and kind of have some clues as to what to be looking for. And already we see pharmaceutical companies using those measures in their trial design. By the way, that might help FDA reach conclusions, but it's helping them make their investment decisions, equally important. And so this area, and I think it speaks to you have to understand what it takes to get over that goal line. And it's not just about chaperoning the treatments and the target approaches. It's about making sure we probably have, I'm going to guess, does this sound right, 35 programs that we're funding in alpha-synuclein alone.
Right.
Okay, 35 different horses. We have to make sure the track runs well. And getting the biomarkers is about the track. Every single one of those programs, we don't know which 35 approaches in the alpha-synuclein portfolio are going to work. We hope all of them work. We assume most of them won't because there's such a high failure rate. But if we don't get the biomarker, whoever's at the front is not going to have the tools they need to go into the clinic in phase 3. And so I think these are things like understanding that soup to nuts, what it takes to get to move that dial and get the treatments we're looking for is really a holistic point of view, and one where we can kind of sit and look at activities all over the field and understand the progress and what's needed. And so I think that mix is what's exciting us, because we do have a lot more things moving forward, much more novel approaches, and we are building out these tools that give us extra confidence that more of them are going to get a better shot.
There's also there's this appreciation over the years that Parkinson's is not just this narrow disease of the dopamine neurons in one brain region. There's other things going on. It's really a syndrome, and other neurotransmitter pathways are involved, other symptoms are involved. So we have active research, and we fund programs also to look at non-motor symptoms. So we're looking at things like constipation. There's some advanced, actually there was a drug that was approved in May of this year specifically for Parkinson's disease dementia. And the idea is that it could be that the mechanisms, the neurological mechanisms that underpin dementia in a Parkinson's patient can be different or distinct from an Alzheimer's patient or different dementias. And so we're, the community, the drug community, the scientists, the researchers are finally getting with what the patients have known all along, which is it's not just the motor, it's not just the movement.
So we are on Parkinson's, we're thinking about it every day. And the ability to go from passive to active is a luxury. And you know, this concept of roadmaps is a very, it sounds like it should be a simple way to think about things, but it turns out science is not built for a single player to kind of step above the field and say, here's kind of, I'm going to consolidate the understandings, the knowledge, bring key people, and we're going to put together a plan. And you got it, that has to be flexible because we're going to learn more things and we're going to change. But just to galvanize everybody is not so easy. And we first got the chance to do this and we became so convinced. It's kind of what you'd like to do, but the ability to do is hard. Our largest donor, and I think the whole Parkinson's field owes a debt of gratitude to this family, is Sergey Brin from Google. And his mom has Parkinson's disease. She happens to be a LRRK2 carrier, and so they didn't know that. That was discovered in the course of her living with Parkinson's and in the course of him being a donor. But when they realized that that was the cause of her specific Parkinson's, and the family's very interested in science and genetics, and we already were, you know, Sergey was already a donor, we went to him and said, would you be willing to help us do something that's never been done? Early in the understanding of a brand new target, can we put together a roadmap? Let's work on biology. Let's build out cohorts, because it takes a while to find who are the people that we can study, both to understand the disease and ultimately be in trials. How are we going to build the right tools for the field so that everybody can, you know, work on the track for LRRK2? And let's go right to companies and see, can we help support them on some early stage LRRK2 projects? And his gift the prior year had, well, they're all generous, but his gift the prior year had been, I think, $10 million. And I said, I think it's going to cost $30 million to do this. And he paused and he came back and he gave us $25 million, said, go crazy on LRRK2. And so first of all, we had $25 million, which means everybody came to every meeting we had about LRRK2. So talk about pulling everybody in. But that group, and it's grown a lot, this ability to put out a roadmap early and get everybody thinking, not what are we going to do in sequence, but what can we do on what ships do we launch on day one, was magnificent. And LRRK2 is going to get to the clinic in record time because of it. And this concept of roadmaps now is really, once something kind of emerges enough, we say, okay, how are we going to get a roadmap formulated around this? And so the ability to kind of now pull together all the things that we've been doing a little at a time and kind of disparately around inflammation, this is exciting work for a Trino to be leading.
Yeah, because it brings together the academics. We have people coming from pharmaceutical companies. We have representatives from Parkinson's UK, because we want to make sure that we're not, it just turns out that they're also interested in inflammation, and so we want to make sure we're not doing the same things. We want to be complementary, and yeah, I'm very excited about it. And just having the buy-in to you know, really just look at the problem and try to be as creative as possible. People think scientists aren't creative. We're just creative in a different way. We just don't translate it well.
Right. And I think these roadmaps are a way that we can define and say, when we talk about that there's $200 million of unmet, you know, again, the science is ahead of the money. If we had more money, we could immediately disseminate it and make some great progress. I think these roadmaps are a way that we're specifically defining that. So one more question for Debi, a bigger picture question for you. I know you and Todd annually prepare for the board, and one of the biggest questions that you have to answer is, what keeps you up at night? And would you be willing to share a little bit about what it is that keeps you up at night, keeps Todd up at night?
Yeah, so Todd is the CEO. I was the founding CEO of the organization, and I stepped out of that role about 8 or 9 years ago, and I adopted twins and moved to Delaware, so I commute to Manhattan from Delaware. And so Todd has risen into the role of CEO. So he and I are kind of partners in crime, and that is true. I guess every 18 months we're asked to talk to the board about what's keeping us up at night. And I'd say I have a couple of things that I zero in on. I stopped worrying about raising the money, because that's always a problem. Like that's normal. I lose sleep every day on that one. But at the margin, I think there are a couple of things I'm always thinking about. One, how do we make sure that our scientific team takes risk. What does taking risk mean? And I think this is a very tricky question to ask and answer, because I've seen us as being the renegades. You know, most people, when they fund research, they ask for your idea and some preliminary data. Most scientists have already done the experiment and they're giving the answers. So the money goes to them because it looks safe, it looks smart, but it's probably not new. And so we took a lot of risk early by doing things like, with very little preliminary data, we were willing to give people money. And that is part of what has seeded the entire new pipeline that we have today. A lot of these things are stuff we put $100,000 into 8 or 15 years ago. And so how do we know that, by the way, when you are using very little data, you better understand the whole field. You better understand all the places in the world where people are thinking about Parkinson's and what is really a new idea. Because believe me, we've had people come to us with things that are truly crazy, and how do you know that that's whether it's crazy or they're onto something, because you don't have much. And so for me, one of the things I fear or worry about is, how do we know that our team doesn't just get in a groove that's easy? And it kind of goes hand in hand with another thing, which is I think 5 years ago we were renegade, but we were also kind of novel and new and small and kind of scrappy. I will tell you, 15 years into it, we are still renegades. We are still scrappy, but we don't look like that to many people. Our staff is 125 people. This year, if we're lucky, we'll get to put about $90 million to work in science. That makes us a significant player. And what's happening is, I worry, how we can't be complacent. So that's a cultural thing. It's a dialogue, an internal dialogue around how do you think about risk, what role should you be playing. But it's also an external thing, that we went from being the David against the Goliath of the traditional, slow, non-patient-centered efforts to now some people see us as the Goliath. Oh, you guys don't care about that. It's like, no. We have a lot of information. It's not that we don't think that new target is interesting, but we have to be rational. We have to be safe. We need to put rigor around testing ideas. And so holding these standards and still maintaining the aggressive posture, to me, is one of the biggest tensions. And I'd say, while there's been a big transformation in getting the patient community involved, it's kind of like we tripled our success. We went from 1% to 3%. Like, the vast majority of Parkinson's patients and families are unengaged. And if we could unlock just a little bit, it makes a significant difference. And to put it into concrete terms, we talk about kind of the aha moment to the drugstore shelf, and one of the key watershed moments is when something proves worthy and has the potential that we're willing to put it into humans, even though it may not be. We have to figure out if it's safe. The clinical process can take 15 years, which is just deplorable. But one of the reasons it takes so long is because we have to recruit people for phase 1, phase 2, phase 3, and we have to run those trials. Biomarkers would help compress how long it takes to get the answers. But we have a problem before we even get to the moment that we start, which is recruiting people. And just as an example, in Parkinson's, if you're designing a trial, you would be lucky to recruit 1 patient per month per site. And so the first phase is smaller and usually in one place, and they'll need about 20 to 30 people. Okay, it takes 20 to 30 months to get everybody into that trial. Phase 2, you might be in 3 sites and you're gonna need 60 people, but 3 sites are only it takes, out of that whole period of phase 1, phase 2, phase 3, something like 6 to 8 years are lost just in recruitment. And I will tell you, you can bring all the smart scientists in the world together, but there are some parts of science that only the patient can participate in. It does not, nobody else can step in and say, I'm gonna be part of those trials. Now, some trials are interventional to test drugs, and I'm the first one to say there's risk in participating in trials. Some trials, though, are observational, where we're just trying to learn. Those should be much easier to recruit up. I'm a subject in the big biomarker study, and in that I've been followed for 5 years. I'm a normal control. A lot of people, my husband says I'm not really normal, but I'm considered a control. And for 5 years I've been in this study. I go twice a year. I've had 7 lumbar punctures as part of that study. And people say, well, why would you do that? And I'm like, because it's important. And they need to know what's different about me changing the last 5 years than someone with Parkinson's disease. And so participating in research, if you could just unlock more of the Parkinson's patient community, when you do double the number of people who get into research, you cut years out of the predicted path for drug development. So this to me keeps me up at night because we don't know how to find folks. Now, I started off by talking about that big footprint that big chapter-based organizations have. We don't have that. That was a conscious decision. We're all in New York City and we're funding globally. And we get out and we meet people, Tricia and others, Liz from our Team Fox group and Axie in the back. I mean, we're out meeting people, but we can't talk to everybody. And so I worry, how are we going to reach more Parkinson's patients and families? How are we going to get them involved in understanding their disease and the treatment options, getting them to the right doctors, and getting them into clinical research? And so that's another thing that I have so much empathy and understanding for, and yet I know that it's an important part of our progress. And so I guess that's another thing. How do we engage more people? So we still, I guess I'll say one last thing, which is actually pretty hard for me to do, it turns out. So this is the best drug development pipeline in Parkinson's in about 40 years. So there is no question that there's a lot to be excited about. But I have to say, the hope versus the hype, it's really important to realize how hard it is to get things over the goal line, and it takes all of us in this room. And so we're really grateful for folks to be engaged and to come out and to care and to help raise money, but also sit here and learn and listen to words like alpha-synuclein and what was the other one, mitochondrial something or another.
Dynamics.
Yeah, and
Dynamics,
so,
yeah.
thank goodness there are PhDs doing this stuff. But I think that there's a lot to be excited about. And so while I'm up at night, I'm also very optimistic.